usually shuts everybody up. A normal curve, okay? So this is on the class webpage. Take a look at it. You'll see where you stand, etc. Any questions, concerns, problems, let me know. I'm happy to talk to you. A lot of energy in here today. It must be the sunshine. Triple shot mocha. I'm standing back. Okay. All right, so last time I introduced the topic of fatty acid synthesis. I'm going to finish that today. I'm going to talk about the prostaglandin metabolism as well. And that should finish this cycle of metabolic reactions relating to fatty acids and their related compounds. Last time I just got a start on fatty acid synthesis. And what I told you, first of all, was that fatty acid synthesis chemically is very similar to the reversal of fatty acid oxidation. And so I'll show you more of those reactions today. But I would also like to point out to you some of the places where fatty acid synthesis differs from fatty acid oxidation. So one of the places in which fatty acid synthesis differs is its location. So fatty acid oxidation, you recall, occurs in the matrix of the mitochondrion. Fatty acid synthesis occurs in the cytoplasm, for the most part. There are some exceptions, and you'll see, I'll point those out to you. But the general fatty acid synthesis that we talk about occurs in the cytoplasm. Okay? Now, when I say general fatty acid synthesis, we'll see that all fatty acids in the cell are synthesized to start with in the same way. They all start and become made into 16 carbon units called palmitic acid, which we've talked about before. Okay? Another difference between fatty acid synthesis and fatty acid oxidation is that fatty acid synthesis occurs not on molecules that contain CoA, but rather a related compound called ACP, or acyl carrier protein. Acyl carrier protein has a component that looks like CoA, but it's not. And so what we're going to see is that both acetyl-CoA and malonyl-CoA are needed to get things started. Not simply to make malonyl-CoA, but we'll actually see that both of these are needed to get things started. Both of these will be converted into ACP derivatives. We'll start with acetyl ACP and malonyl ACP. So we have to make a malonyl-CoA first before we can make a malonyl ACP. Okay? So ACP is the carrier for fatty acid synthesis. Let's take a look now at what happens in making fatty acids. Okay? So, as I said, we have to start with an acetyl ACP and a malonyl ACP. So we need both. You'll notice that these things are color-coded so that we can see where the growing fatty acid chain is. Okay? Well, what happens, it turns out, in this process, we see the acetyl ACP here, the malonyl ACP here. We see that the new carbons that are being added, in other words, the red guys are the new ones, they will remain linked to ACP. The previous one will get linked to what would be carbon number 2. So we see the blue guys here coming on to carbon number 2. The newest ones are at the ACP end. When we go and we elongate this one, then these will be added to another ACP, and we'll talk about that as I get there. Okay? So the newest carbons will always be on the end closest to the ACP. Now, let's look at what happens in this process. First of all, we are starting with a 2-carbon molecule and a 3-carbon molecule, acetyl ACP and malonyl ACP. And we're adding those 2 carbons, all right, by cutting off a carboxyl group. So the very first thing that happens is this guy here attacks this bond, resulting in the decarboxylation and the joining of the blue to the red. Okay? So we've made a condensation reaction. Now that's not the exact opposite of what we did in the thiolase reaction where we split it off. Okay? Because when we split it off, we didn't come and bring back in an extra carboxyl group. So we don't have the exact reversal here. The other reactions that we'll talk about are pretty much the same reversals. Well, at this point we have acetoacetyl ACP. It has 4 carbons. Okay? And these 4 carbons look very much like what we saw during fatty acid oxidation. In fatty acid oxidation, we took and we broke this bond, made two acetyl-CoAs. The step before this one involved an oxidation that converted a hydroxyl to a ketone. Okay? In fatty acid synthesis, we're going to go backwards. We're going to convert that ketone back to a hydroxyl. You'll notice that the configuration of that hydroxyl is in the D configuration. In the oxidation, it was in the L configuration. Okay? But you'll notice also that this hydroxyl is on carbon number 3, just like we had during fatty acid oxidation. Okay? Exactly as we saw. Notice that the source of electrons is NADPH. You haven't seen that yet. Well, we saw it briefly the other day. NADPH is an electron carrier, usually for biosynthetic reactions. So when we're making something, the source of electrons is usually NADPH, not NADH. That sort of makes sense because NADH's electrons go into electron transport. Having a different source of electrons for synthesis makes sense, and that's why NADPH is important. So NADPH is the source of electrons for that reduction. In the next step, and by the way, I'm not worried about these names. Okay? I think if you know the generalities of what's happening, right? We're going from a ketone to a hydroxyl. That's occurring on position 3. You know that's a molecule that has a D configuration, not an L configuration. The name to me is not nearly as important. Okay? All right. In fatty acid oxidation, we produced that hydroxyl by adding water across a double bond. In fatty acid synthesis, we remove water to create a double bond. So again, we're doing the basic chemical reversal of what we saw in fatty acid oxidation. We're removing a water. You'll notice that we create a trans double bond just like we saw in fatty acid oxidation. You'll notice it's between carbons numbers 2 and 3, just like it was in fatty acid oxidation. So again, reversal of that general chemical process that we saw before. Well, in the last step on this round of synthesis, we have to reduce that double bond to a single bond. In fatty acid oxidation, you recall we went from a single to a double. We had to pull electrons away. In this direction, we have to add electrons. And again, you see that the electrons are coming from NADPH. Okay? Now, you'll notice I didn't give you any enzyme names there. And there's a very good reason. Fatty acid biosynthesis has a giant complex of enzymes, each of which have massive names that we're not going to give individual Wallace and Gromit names to. Okay? We'll give it a very simple name. Okay? And I'll show you that name as we get to it. So this is the end product of one round of fatty acid synthesis. Now notice I said one round because when I started this lecture, I said that what fatty acids are doing is they're being made into 16 carbon units. This clearly doesn't have 16 carbon units. So what we're going to do on the next round is this will be our starting material. Instead of being acetyl ACP, it would be butyryl ACP. This would add on to the new carbons coming in from malonyl ACP. Okay? And when we finish that round of synthesis, we'll have a fatty acid that has 6 carbons. And we continue synthesizing until ultimately we get to a fatty acid that has 16. That finishes the basic synthesis, or the simple synthesis, of a fatty acid. So getting to 16 is the simple synthesis. It's for this reason that palmitic acid, which has 16 carbons, is the most abundant fatty acid in our bodies. Okay? Now, there are things that happen to the fatty acid after it's at 16. I'll talk about those. But before I talk about those, I need to make sure we understand how we're getting to 16. So this cycle continues and continues until we get to 16. All right. Now, here are the individual reactions, blah, blah, blah. And here are the names of the individual. It says enzyme. What they really mean is activity. Okay? Because with the exception of the acetyl-CoA carboxylase, all the other guys are located in a complex. Remember we had the pyruvate dehydrogenase complex where we had 3 different catalytic activities in that complex? E1, E2, and E3. Okay? This complex has all these activities, and we're not going to memorize those names. This complex name is called fatty acid synthase. Very simple name. So all of these reactions after the acetyl-CoA carboxylase are part of the fatty acid synthase complex. Now, the beauty of having this in a complex is that all of these reactions can occur right next to each other very readily. And you'll notice I'm drawing my hand in a circle. It turns out this enzyme has a reaction scheme that's almost like the hands of a clock. It points part of the molecule over here, something happens, it points it down here, something happens, it points it down here, something happens, literally going around in a clock-like fashion. Okay? And only after this thing has gone around enough times to make 16 carbons, I think you'll count, you'll get 7, okay? 7 times around, when it gets to 16 carbons, the 16 carbon molecule no longer fits in the clock and it's released. So that's why the basic synthesis or the simple synthesis only occurs getting up to 16 carbons. Okay? Make sense? Yes?
Does it always end in palmitoyl-CoA, or are there other 16 carbon?
Okay. Does it always end in palmitoyl-CoA? It ends in palmitoyl-ACP, and then of course the ACP would be swapped out for a CoA. There are things that can happen to that, and I'm going to talk about those things as well. But once it's gotten to the 15 carbon state, basically that ACP is removed and a CoA is put onto it. So yes, the answer to your question, but other things can happen to the CoA version.
After.
That's right. And I'm going to talk about those as well. You'll like the synthesis because it's simpler than the oxidation. Okay. Other questions? Okay. So fatty acid synthesis. And by the way, this says in bacteria, you have the very same complex in your cells doing the very same thing. They're all very, very similar. The schematics that your book uses are almost bizarre, so I'm not going to go through that. Okay. One of the things we need to discuss is, well, how do we get fatty acid synthesis going on in the cytoplasm when our acetyl-CoA is in the mitochondria? All right? Well, if you remember back, you might wonder, well, why isn't the mitochondria in the first place? Let's think back to what's happening in the citric acid cycle when we're not exercising. What did I say happened to the citric acid cycle when we stopped exercising? So ATP concentrations go up because we're not burning it. Oxidative phosphorylation stops. Proton gradient gets high. Electron transport stops. NADH concentrations go up. Citric acid cycle stops. All right, where is it going to stop? Well, it's going to stop at reactions ahead of, that is, the ones preceding places where NAD is needed. Okay? Isocitrate concentrations will go up. Citrate concentrations will go up. And if you think about it, when citrate concentrations go up, what do we use to make citrate? Acetyl-CoA. So we're going to have a lot more free acetyl-CoA sitting around when we're not burning up ATP. So we've got two problems. One problem is that citrate is going to tell acetyl-CoA carboxylase, get active and start using fatty acids. Okay? Second thing that's going to happen is acetyl-CoA concentrations are going up, so the cell is going to have plenty of material to make fatty acids. So these work hand in hand to ensure that you make fatty acids when you are not burning up all of your ATP. All right. Well, again, we're thinking about the mitochondrial matrix, but the action things are out in the cytoplasm. So how do we deal with that? Well, it turns out, not surprisingly, there's a shuttle that moves citrate when it starts to accumulate from the matrix out to the cytoplasm. This is an antiport. Citrate is swapped for pyruvate. Pyruvate comes in, citrate goes out. What happens to pyruvate when it gets in here? Well, this shows it going to oxaloacetate. It could just as easily go to acetyl-CoA, which is going to be a problem. All right? Citrate comes out here into the cytoplasm, and what happens to citrate? It gets broken down by an enzyme called citrate lyase. Now that's different. It sounds like the enzyme that we talked about in the glyoxylate cycle, which was isocitrate lyase. They're not the same thing. Citrate lyase cleaves citrate into oxaloacetate and acetyl-CoA. There's the acetyl-CoA for your fatty acid synthesis. And if you don't break all this down into that, what happens to citrate concentrations? It goes up and activates acetyl-CoA carboxylase. So we can see now how these guys are working together to stimulate synthesis of fatty acids in the cytoplasm when you're not burning ATP. Okay, makes sense? Questions? Okay. Let us proceed then. Excuse me. To talk about, I've already talked about the control, so I'm not going to go through that there. I will talk about how we make unsaturated fatty acids. And this looks really hairy. All right? Let's start with the name of the enzymes involved in this process. There's one main enzyme we'll be concerned about. We give it a nice, simple name. It's called a desaturase. A desaturase uses all the reactions up here, which we're not going to memorize. We're not going to memorize the reactions up here, because for our purposes, the main thing that's important is that they're making unsaturated fatty acids. So desaturases are essential for that. This process occurs in the endoplasmic reticulum. In the endoplasmic reticulum. Okay? Now, what's interesting about this is that mammals, you may recall, cannot make double bonds farther away than the 9th carbon starting with the carboxyl group. Okay? So that 9th carbon, we can make a double bond there. If we need double bonds, let's say we've got something that's 18 carbons long and we need a double bond at 12 or 15, we can't synthesize that using our desaturases. Our desaturases will not work beyond carbon number 9. And as a consequence, when we need fatty acids like linoleic or linolenic acid, those are called essential fatty acids, meaning we have to have them in our diet because our desaturases will not make them for us. If we look at other organisms, they have desaturases that will, in fact, make double bonds further out there on that chain. Okay? So desaturases are different depending upon the species that they come from. Mammals do not make it beyond position number 9. Okay. Here are some unsaturated fatty acids. We talked about fatty acid structure before, so I'm not going to go through those and show them to you. But I do think that you should know some basic fatty acids. Linolenate, which is the ion of linolenic acid, that is an essential fatty acid. It has double bonds at positions 9, 12, and 15. We need this. We can't make it. Linoleic acid, and you'll notice the omega designation. Remember the omega designation? Instead of counting from the carboxyl, we start counting from the methyl end. So because this guy has 18 carbons, and its last double bond is at position 15, you can figure omega-3 means it's 3 from the methyl end. Okay? Linoleic acid has double bonds at positions 9 and 12. It also has 18 carbons. And since if we subtract 12 from 18, we get 6. This is an omega-6 fatty acid. Linoleic acid is also an essential fatty acid. Okay? Palmitoyl-CoA acid is a 16-carbon fatty acid, as its name suggests, palmito meaning palmitic acid. It has omega-7. All right? So if it's omega-7 and it has 16 carbons, you can figure that double bond is at position number 9. We can make this one. Okay? We can make this one. Here's oleic acid, 18 carbons. Omega-9, 9 from 18 tells us we have a double bond at position 9. We can make this one as well. So the top 2 are essential. The bottom 2 are not essential. Make sense? Yes?
On that last figure you had for the desaturase, what ultimately is accepting electrons in double bond?
I'm reluctant to tell you because it's a very complicated scheme. Let me show you why I'm reluctant to tell you and why it's a complicated scheme. Okay. Ultimately, the electrons are moving from NADH to NAD, which looks like a donation of electrons. Okay. So you see this swap, 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 swap that ultimately results in the removal of electrons down here. So I think rather than confuse the class and go through that this time, if you want to come talk to me, I'll talk to you about that separately. Because it looks like, whoa, it should be going in, not out. Okay? Okay. That's pretty much... Oh, so, and there's one other set of enzymes, I don't even have it on here, I need to tell you about, that's important for fatty acid biosynthesis. And these fatty acids are, for example, I've told you how you get to 16 carbon fatty acids, but I haven't told you how you get beyond 16. Okay? So this requires action of another group of enzymes known as elongases. Elongases catalyze the addition of carbons to positions beyond position 16. Now these guys are found, interestingly, in two places. One is the endoplasmic reticulum. That's not totally surprising. That's where we saw the desaturases. Okay? The other place that we see them is rather surprising, and that is we find them in the mitochondrial matrix. The mitochondrial matrix. Now, that's surprising because remember what's going on mostly in the mitochondrial matrix is beta oxidation, the breaking down of fatty acids. A real good exam question. You know, once I say it's a real good exam question, it probably won't be an exam question. But a real good exam question, or maybe I should leave it as an exam question. No, we'll leave it as a class question. A real good question is, how does the cell manage that? How does the cell keep straight, whether it's synthesizing or whether it's oxidizing? And by the way, it's CoA. Once we've made palmitoyl, it's not the ACP versus CoA. They both have CoAs. How does the mitochondrion know what it's making and not what and what it's breaking down?
Energy levels?
The what?
Is it energy levels?
Not energy levels. Nice try. You guys actually know the answer, but you're not thinking about it. You will. Yes?
The amount of acetyl-CoA in the...
Is the amount of acetyl-CoA in the mitochondria? Nope. How about Joe?
Is it the long chain form of acyl-CoA dehydrogenase in the peroxisomes and not the mitochondria?
Exactly. So remember the acyl-CoA dehydrogenase, we have a long form, we have a medium form, and a short form. Where was the long form? Longer than 16. It was out in the peroxisomes, right? Okay. There's not an enzyme there that's going to touch it to start. That's why when that gets in there, it says, oh, you want to make this longer, you're making it longer. It's an odd scheme. It's a very odd scheme, but that's the way the cell is set up to do. Danielle?
If there are fatty acids shorter than 16,
Yes?
does it just stop when it gets to that, or does it make 16 and they get taken off?
Okay. So I'm not sure I understand the question, but basically, cells will not make fatty acids shorter than 16. Okay? With that complex that I talked about. Basically, they're making 16 carbon fatty acids at that point.
Are there fatty acids that have less than?
There are fatty acids that have less, and they have other considerations in their synthesis.
So they might be made somewhere else, or
They might be partially broken down. Yeah. Okay? Now, shorter than 16 is not real common. Okay. Now, so the cell really has a nice way of keeping track then, whether I'm going to oxidize or synthesize. Why that's in the mitochondrial matrix, I don't have a good answer, but I can give you a thought. Okay? Maybe I should ask you to give me a thought. Why do you suppose it might be useful for the cell to have this activity in the mitochondrial matrix? Shannon?
Because the mitochondria is so selective that there's a ton of fatty acids out in the rest of the cell. So if you have that elongated thing, it gets free stuff out of the cell.
Okay, so she said that the sort of mitochondrial is more selective, is what I remember. Basically, no. Okay, so well, no. I'm asking for your opinions. No, it's a nice guess. I don't mean to dump on it. But there's another reason that and by the way, there's not an answer to the question. Okay, so I'm asking you to speculate. Your speculation is as good as anybody's. So that wasn't what I had in mind. Maybe that's what I should say. Yes, sir. Ample acetyl-CoA. Where might that be a factor? How about when you stop exercising? When you stop exercising, what happens to acetyl-CoA concentrations in the mitochondrion? It goes up. I've shown you how you can run it out to the cytoplasm. Here's another thing that you can do with acetyl-CoA. You can put it onto fatty acids that you've already got in there, make them longer, and simplify the process of fatty acid synthesis. Instead of exporting them out of the mitochondrion, going and doing something, and then turning around and bringing them back in, it doesn't make any sense. Why not just make them longer when we've already got them in there if we have plenty of acetyl-CoA around? Does that make sense? You like that explanation better or not? Okay. You get an A. All right. Other questions, comments? Okay, this is a good place for a song. Let's do a song. And what's that? This song is to the same tune. By now you guys ought to know the tune very well. You would think I was in a rut with this, but in fact, the 3 songs that I have about this were written in very, very different periods of time, over a span of years. Just coincidentally, they all happen to converge right here. This is called When Acids Are Synthesized. So, the 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within deoxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with the glycerol, so you get obese when acids are synthesized. Okay. Excuse me. I promise it's the last time we'll use that tune this term. Although you guys are getting good with it by this point. So, all right. Well, we're moving along fairly well, and we'll actually finish early today. One of the things that we will talk about over the rest of the period is the synthesis and action of a very interesting group of compounds called prostaglandins. I referred to them in class 2 or 3 times before, and I promised you that we would learn something about their metabolism, and that's what we're getting ready to talk about right now. Okay? Now, prostaglandins are molecules, okay, that have 20 carbons, and they are produced in the body from the 20-carbon polyunsaturated amino acid known as arachidonate or arachidonic acid. Okay? Arachidonate has 20 carbons. It has 4 double bonds. Okay? And arachidonate is found in glycerophospholipids. Okay? That's where we find it. We don't find hardly any free arachidonic acid around, either as arachidonic acid or as arachidonyl-CoA. The place where we find it is in the glycerophospholipids of the membrane. And there's a very good reason we don't see any of it free. Whenever we have it free, we convert it into prostaglandins. Okay? Now, prostaglandins have many different functions. They range from causing inflammation and swelling and pain, okay, to functions of uterine contraction in childbirth. They may cause menstrual cramps. They may affect blood pressure. They may affect the blood clotting. They have many different things. And as a consequence, there are many different prostaglandins. Some do one thing, some do another thing. Some of them seem to work counter to each other. Okay? Some of them work counter to each other because some of them can actually help to facilitate blood flow instead of clotting blood flow. So there's some very almost contradictory actions that some of these compounds can have if we compare one versus the other. We're not going to compare them. Okay? Now, these compounds, as I said, are made from arachidonic acid. All right? Arachidonic acid, most common source of that is from glycerophospholipids. Okay? You see diacylglycerols, you say, well, how about there? Well, these are on their way to becoming glycerophospholipids, or they were originally a glycerophospholipid for which the phospho part got removed. So ultimately, glycerophospholipids are producing arachidonic acid. Now, this PLA2 here is an enzyme called phospholipase A2. That's the enzyme that cleaves arachidonic acid from the glycerophospholipid in your cell membrane. Okay? Now let me give you a scenario where this will happen. Okay? A bee stings you. A bee stings you. It makes a compound called melatonin. Melatonin stimulates the action of this enzyme, produces arachidonate. Arachidonate is very readily converted into prostaglandins. And at that site of the bee sting, you start producing swelling and inflammation, which is why a bee sting hurts. Okay? That arises because of activation of this process right here. Okay? Now, I was very curious one time. I used to have a big garden out in the country, and I thought, well, before I tell that story, I got to tell you one more thing. You get old, you mix your stories up, you know. So, I'm getting old. All right. So, another thing I need to tell you is that this enzyme here called prostaglandin synthase is responsible for making prostaglandins, no surprise. It has a couple of other names. One of the other names is it's called a cyclooxygenase. I'll show you why that's the case when I show you structures in a minute. Okay? We don't need to worry about structures for the moment. Cyclooxygenase are abbreviated COX, C-O-X. They're called COX enzymes as well. And it turns out there are at least 3 and maybe 4 different classes of COX enzymes. All right? Now, I'll tell you something about those classes in a minute, but to get back to my story I was going to tell you, COX enzymes are inhibited by aspirin. Okay? One of the reasons aspirin is a painkiller is it's reducing the production of prostaglandins. Okay? Now, aspirin has a lot of effects. All right? Reducing the production of prostaglandins reduces the pain associated with them, headache, maybe the bee sting, maybe whatever. Okay? However, aspirin also has some other side effects. One of the side effects is that people have sensitivity in their stomach. Some people are very sensitive to aspirin. And they used to say, well, it was because aspirin was so acidic, et cetera, et cetera. The answer is not because aspirin's acidic, but rather because aspirin was inhibiting some prostaglandins that were needed for maintenance of intestinal tissue. So in some people, this doesn't happen in everybody, but in some people, when they take aspirin over a long period of time, their stomachs get very, very irritated, very, very sensitive. And the reason that's happening is because they're inhibiting production of a prostaglandin that's needed to maintain that tissue. Okay. All right. So this enzyme works very rapidly. Okay? A bee stings you, it doesn't take very long, you're swollen. So my story I was going to tell you was, I had the idea one time that what if I took aspirin almost immediately upon getting a bee sting? Okay? Would it work? Would it help? Okay, well, we lived out in the country and I had a garden and I was every now and then getting stung by bees. Okay, so I thought, well, the next time I get stung by a bee, I will try this. So I'm working out in the garden one afternoon, and all of a sudden, kapow, I get it right here. And the first thing I think of wasn't, damn, let's get rid of this bee. It's, let's run inside and go grab some aspirin. Okay? So I went in literally with the bee still sticking inside of me. Okay? Grabbed an aspirin, or a couple aspirins, and gobbled them down. I had it down within, I don't know, maybe 10 seconds of getting stung. And Dara's wondering what in the heck I'm doing, you know? Get out of my way, I need an aspirin. I can make an inappropriate joke at this point that I won't. But so I needed to have this aspirin right now, and so I go in, I get the aspirin, and I'll be damned. The swelling, I usually swell up pretty good size with bees. Okay, the swelling really didn't take place very much. So I had to get it in my system very quickly. I had to get it into my bloodstream very quickly, but it seemed to reduce the severity. Okay, so that was my only data point, my only experiment in this process, but it seemed to work. Okay, now, yes, Danielle.
So you said some of these prostaglandins are involved in clotting blood?
They're not involved in clotting of blood, but I'm just getting ready to talk about that. Let me talk about that a little bit, okay? So prostaglandins do seem to play a role in what's called the stickiness of platelets, cells in the bloodstream. The stickiness, how much they clump together. This appears to play a role in the clotting process itself. The stickier they are, the more easily you clot. And especially for people who are prone to clotting. Yes, ma'am?
Is that why if you take aspirin, it thins your blood?
To do what?
It thins your blood.
Yes, so I'm explaining that thinning mechanism. So she's reading my mind, okay? So prostaglandins, okay, if you reduce the production of some prostaglandins, you reduce the stickiness of those platelets. And so just as she said, some people take this to do what's called thinning of the blood, and we talk about thinning the blood with prostaglandins. It's different than the warfarin thing we talked about last term, okay, which was affecting vitamin K metabolism, or vitamin K action, I should say. In this case, we're actually reducing the stickiness, which is a different component of the clotting mechanism. So one of the things that your doctor will ask you if you ever go in for like surgery or something like that is to make sure that you're not taking aspirin within a few days of surgery, because they really do want your blood to clot. So yes, the answer to that is yes. Okay? Yes?
And so that's why people would take it for heart attacks? Although they are aspirin.
Okay, and so the reason people take it they have concerns about heart attacks or stroke, in either case, yes, exactly that. They will take that so that they're reducing the stickiness. And again, you have to be careful. You don't want to take too much, because you do need to have a certain amount of clotting. Like we said last term, if you take too much warfarin, what happens is you die, not because you all of a sudden get all this blood coming out of you, but because you hemorrhage. And so hemorrhaging in the brain is not good. So you want to be careful in managing that. Okay? The typical dose for an adult that has been shown to have some benefit is about a half an aspirin a day. And your physician will always say, talk to your doctor before doing this first. Yes, sir?
If you're going into surgery and you've been taking aspirin, or if you get in a car accident, how do they reverse that?
Okay, so if you go in, let's say you have a car accident and you've been taking aspirin, how do they deal with that? Well, one of the things that hopefully you're doing is you're taking aspirin that is of the proper amount. So half an aspirin a day reduces stickiness, doesn't have a major impact. Ideally, you wouldn't be taking that aspirin if you're going to have surgery. You can't plan a car crash. If you were taking a lot of aspirin over a longer period of time, that would be a bigger concern, and they would no doubt have to give you a lot of more blood in transfusion during a surgery. Let's say you were in an accident or something. So the blood, remember that the blood that they're giving you will not have that same treatment. So you would clot, but it would just be more of a problem. Make sense?
Yeah.
Yes, Tamsin.
If half an aspirin is the dose, why do they give you a whole 2 doses of this?
If half an aspirin is a dose, why do they give you 2 doses? Well, the original dose of aspirin had nothing to do with this. So there are some that are made specifically for that purpose, and you don't have to cut it in half. But the original dosage of aspirin was to reduce pain and so forth. So that's the common size that's made. Okay? Yes?
I hear about real specific elongases that are inhibiting. So where are we actually inhibiting this process with aspirin?
You hear about what?
I hear about specific, very specific
Yes.
prostaglandins that are inhibited.
Okay.
So where are we actually?
Okay, so let me show you. It's a good question. Let me show you the structures and show you where this is happening, and then tell you what's happening. So that when we see structures, it's going to happen. Before I do that, I just want to point out a couple things. So, one, we can inhibit synthesis of prostaglandins from arachidonate with aspirin. Aspirin is called an NSAID, non-steroidal anti-inflammatory drug, NSAID. Okay. There are other NSAIDs. They include ibuprofen. Okay. They include some other unusual ones that aren't really much in the market anymore, including things like Vioxx, and there's a couple of others. Okay. I'll tell you something about those in a minute. The other place that we can stop the synthesis of prostaglandins is by inhibiting this enzyme right here, PLA2. To inhibit this enzyme, we have to use steroids. They're called steroidal anti-inflammatory drugs. And they work by inhibiting the release of arachidonic acid from the glycerophospholipids. If we stop the release of arachidonic acid, therefore, we stop the production of prostaglandins. Ideally, we don't want to be taking steroids. Ideally, if we can stop it here, we want to. However, if you have a severe situation with swelling or inflammation, you may be put onto steroidal, like corticosteroids, to help reduce the action of these enzymes. Now, there's two other classes of compounds here that I want to talk about. One, the leukotrienes. They're also made from arachidonic acid. Leukotrienes have roles also in things like mucus production. They also regulate things like your bronchial tubes in your lungs. Leukotrienes have roles in asthma. Some of the strategies for treating asthma are to use leukotriene inhibitors, and some of these, at least, are promising. Okay? Prostaglandin, you'll notice this is called H2. That's the primordial prostaglandin. There are a whole bunch of others that are made from it, but they come through H2. H2 can be made into another class of related compounds called thromboxanes. And these guys also play roles in clotting and stickiness. So again, by stopping this pathway here, we stop production of these, and we result in the thinning of blood. Okay, did you have a question, Daniel? Yeah.
So the leukotrienes are involved in asthma, and some asthma medications, or I was told that they're steroids of some kind.
Mm-hmm.
So is it the same with steroids
Right.
that are used right there, or are there different steroids that stop it in a different process?
That's a very good question. You're talking about things like in inhalers and so forth. And the steroids that are used there are bronchodilators. And I don't know if they're the same ones here or not, to be honest with you. That's a good question. I'll look that up. I don't know the answer to that. Okay. Well, to try to get back to structure of these guys, the prostacyclins we're not going to worry about. We're not going to talk about those.
Most of them are prednisone.
Okay. Okay. So the answer, at least one answer, yes. So, all right. This shows the structures of some individual prostaglandins, or actually one prostaglandin, and some related compounds. Now, we're not going to draw structures, all right? But there's some features of the structure that are structures that are on the screen that I think you should know. First of all, these guys are called eicosanoids. E-I-C-O-S-A-N-O-I-D-S. Eicosanoids derive their name from the fact they have 20 carbons. Eicoso meaning 20. Okay? The 20 carbons all come from arachidonic acid. Now, arachidonic acid starts out with something that has a carboxyl at one end. It goes on and on and on and on. And it has a methyl group at the other end. Okay? One of the things that happens in the prostaglandin synthase, that is the COX enzyme, the cyclooxygenase enzyme, what that enzyme catalyzes is the formation of this bond right here. Okay? It catalyzes the formation of that bond right there. That bond does not exist in the fatty acid because it's a linear chain. This guy puts a bond, makes that 5-member ring, and the reason it's called a cyclooxygenase is it's making a cyclic ring, and that ring will always have at least one oxygen out there. So it's a cyclooxygenase. Other things can be put on there by other enzymes. Here's prostaglandin E2 that has a hydroxyl group out here, etc. Okay? This guy originally had 4 double bonds. It ends up with 3 double bonds as a result of this cyclization that happens. So as I said, arachidonic acid has 4 to start with. Okay? Thromboxane is made from prostaglandins, and so you can see, not surprisingly, that thromboxane would have a ring, and it would have at least one oxygen. You can see it's been altered out here, but it has at least one oxygen out in the ring. Prostacyclins we're not going to talk about. Leukotrienes, you recall, are not made from prostaglandins. They're made from arachidonic acid, and notice, they don't have this bond. They're not going through that COX enzyme. They're going upwards instead of downwards. Okay? So if I were to ask you the fundamental difference between a leukotriene and a prostaglandin, I would hope you would recognize it doesn't have the cyclic, the ring structure that's made by the prostaglandin synthase. Okay, now I want to finish talking, telling you a story which hopefully will address your question a little bit about the different places of synthesis and so forth of the different prostaglandins. There are dozens of prostaglandins, and we're just not going to have time or the ability to go through the synthesis of each of them. And their synthesis is a little confusing. Okay? The part of the confusion arises from the fact that there are different COX enzymes. Okay? So COX enzyme, the two most common are called COX-1 and COX-2. COX-1 is fairly abundant throughout most of the cells of your body. Okay? You have the ability to make those prostaglandins pretty much anywhere that you need to. COX-2 enzymes appear not to be made in most cells of the body, but where they were originally discovered and characterized were in joints. And so one of the reasons that people take aspirin who have, for example, rheumatoid arthritis, have a lot of joint pain, they're taking aspirin or nonsteroidal anti-inflammatories so that they're not making prostaglandins in their joints. But the side effect of those, if you recall, I said intestinal problems, maybe some other issues associated with those. So people who take those over a long period of time were having real problems with intestinal issues. Okay? Well, when they got looking, they discovered that COX-2 enzymes were not being made in intestines. Okay? And that a great search went on to find COX inhibitors that inhibited COX-1, but not, inhibit COX-2, but not COX-1. Okay? So if you inhibit COX-2, what people felt at the time was you will have the perfect pain medication. All right? Because joint pain and so forth you could treat, you would not have the effects in the stomach, et cetera, et cetera. And so billions of dollars were invested by the pharmaceutical companies into creating compounds like Vioxx. That was one of them I mentioned. Okay? Those compounds made it through trials. They made it to market. And then it turned out that they didn't know at the time, or although there was allegations that drug companies hid it, that some of the COX-2 enzymes were necessary in the heart. So they had some side effects, not a large number, but they did have side effects of people having issues with heart as a result of taking the NSAIDs that were COX-2 inhibitors. As a result of that, billions of dollars of investment basically went down the drain because a lot of those were pulled from the market because of the concern of lawsuits and legitimate health concerns with respect to having damage on the heart. Okay? So that was a big turnaround for the drug industry as a result of that. I hear shuffling. So I guess that means perhaps we will save the song for next time then. I have one more song, but we'll save it for next time. You know, I don't know exactly. I don't. The problems that came up with it were associated with some valve problems and other issues with the heart. So you would guess it maybe has something to do with maintenance again. But, yeah.
What?
What's that?
Inflammation of the valves, maybe?
Could be. Yeah. Sure.
You know how you're talking about with the aspirin in the stomach with the prostaglandins?
Yes.
Why is aspirin, I guess, even like a